In this video, we will discuss about this in vivo efficacies of the antibody and targeted protein degradation based nanoparticles. So in this case, initially what they use is this breast cancer, that is MDA MB231, as it has the high expression of this EGFR on the cell surface of the tumor cells. So they inject the mice with this breast cancer tumor cells and then use the antibody and TPD nanoparticles for the treatment. And what they are able to see is that in terms of the tumor volume, they are able to see significant reductions in the tumor volume when both the antibodies and nanoparticles, the targeted protein degradation nanoparticles, is used. That can be evident from the figure B. After that, they also check the expression levels of the EGFR and the apoptosis marker, that is cleaved caspase 3, using immunofluorescence image, and as expected they are able to see a decrease in these EGFR signals. At the same time, they are able to see an increase in the this cleaved caspase 3 signals, indicating that there is an increase in the apoptosis and there is a decrease in this EGFR expressions. EGFR expressions they have further quantified using the Western blot, which is shown in the panel D, where we can see that EGFR expressions is significantly lower when both antibodies and the targeted TPD-NPs are used together. That is in the last column. After that, in order to check any negative effect or side effects, they did the biochemical analysis of the liver and the kidney, and they are able to see a minimum changes as they checked some of the biomarkers for the biochemical changes, and they are only able to see a minimum changes, indicating that there is a minimum side effect of using this drug on the mice. Afterwards, they did their studies on mice with another tumors, that is U87MG glioma tumors. So they inject the mice with these tumors and then use their drugs and use their antibody and TPD-NPs for the treatment. And in this case also they are able to see that this EGFR signals significantly goes down when they use both the TPD nanoparticles and the antibodies targeting EGFR. And they also check the survival probability in this case, and they are able to see the survival probabilities are higher when both antibodies and TPD-NPs are used compared to the case where either antibodies is used or either TPD is used, and in the also compared to the control case where nothing is used. The reason these results are important compared to the other targeted protein degradations nanoparticles, because the other traditionally available approach of this TPD-NPs does not have in vivo results, does not have compatible in vivo results. We can say that because especially for the solid tumors, because of the immunosuppressions that is observed by provided by this tumor microenvironment, that prevents the interactions of the immune cells with the tumor cells. So this process, as they directly target the tumor cells and then use the tumor cells protein recycling process to degrade this targeted protein, and this can overcome the immune evasions that are provided by the immune suppressions, evasions that are provided by the tumor microenvironment of the solid tumors. So this is how they presented that their TPD-NPs are novel compared to the traditional NPs, and it can increase the tumor diagnosis when combined with other antibodies that is also used to target these target proteins, such as in this case they have use the antibodies that can target EGFR. At the same time, they have also prepared their targeted EGFR targeted protein nanoparticles. So the combined effect shows a enhanced tumor suppressions compared to the individual treatment. So this is how they ended their paper by commenting that their TPD-NPs that they have designed, or they designed TPD-NPs as enhanced efficacies against the tumors when combined with other antibodies. So that's all for this video. If you need to understand more about this topic or this paper in detail, you can go through the reference that has been provided in the description. Thank you for your time.